I'd like to take the opportunity to introduce you to the host of today's webinar, and that's Dr. Shemat Joseph. He's an assistant professor of entomology in the Department of Entomology here at the University of Georgia, and he's located with us on the UGA Griffin campus. His research addresses lots of existing, emerging, and invasive pest management issues of both turfgrass and ornamentals. He has projects that aim to enhance our understanding of the biology of both pest and beneficial species, and his major thrust is in developing management tactics that would reduce the impact on both the environment and non-targets. So Dr. Joseph, I'd like to welcome you and introduce our first speaker for today. Dr. Joseph, the floor is yours.
Our first speaker is Dr. Alejandro Del-Pozo. Dr. Del-Pozo is an applied insect ecologist at Virginia Tech. He is located at the Hampton Roads Research Station in Virginia Beach. Alejandro is also an extension specialist serving the ornamentals and turfgrass industries. Alejandro's research is focusing on providing applied and science-based solutions to implement IPM program. Alejandro holds a master's in entomology from Washington State University and a doctoral degree in entomology from North Carolina State University. Alejandro, floor is yours now.
Go Pack, even though I'm a Hokie now, but hey, it is what it is. Good afternoon, everybody. Thank you so much for that introduction, Shemat, and I'm so happy to be here. I'm going to start sharing my screen. All right. How's that looking? It look good?
Looking good.
All right, perfect. All right, thank you. Again, thanks for the invitation. I want to take this opportunity to talk to you all for the next 45 minutes what we see in here in Virginia. And we can use this as a model system for any of you out in the field or doing your activity. So you can learn about what we're doing here, and maybe some of this will resonate with you. And hopefully we can have a nice conversation, so to speak, questions at the end. So please feel free to use the Q&A chat box to drop that at any time. So I decided to present anything that's, you can read the title. Red-headed flea beetle is one of the top pests for open field nurseries here in Virginia and also in Georgia. And it goes all the way from that emerging pest to the newest invasive species for ornamentals, which is the box tree moth. So we're going to talk about that too. So on the title slide, you can see damage from the red-headed flea beetle, that defoliation that nobody wants to see, and growers are really going after that. And on the right, that's Japanese maple scale, like all the layers for the adults with the waxy coating that it's getting there, and then it has some problems. I want to introduce my program. So I'm leading the Applied Insect Ecology Lab here at Virginia Tech. I've been in my position 3 years. So I'm trying to do several research projects within the biology and the ecology serving the green industry. So that's ornamentals and turf. I'm really interested in doing the life history, the phenology. I'm using remote sensing now, all these new cool toys. I'm going to show you a little example about that close to the end of the presentation. But I'm also really interested in planning insect interactions. And then on top of that, knowing all the biology, ecology, we want to propose to end users like you guys or growers or anybody just to propose those control tactics. We try to focus on biological control. There's some cultural practices, but we all know that insecticides are a big part, and we trying to understand what are the most efficacious one. So for the presentation today, I am summarizing what we see in Virginia as a whole state. On the eastern part of Virginia, we have big issues with the red-headed flea beetle. That is a hydrangea on your left-hand side. Across central Virginia has a problem with the Japanese maple scales. I'm going to talk about that too. And then across the whole state, ambrosial beetles, which are these teeny tiny beetles that tunnels and create these toothpicks, like the excrement gets all, and you can see those toothpicks hanging from the affected trees. I'm going to present a little bit of information about that beetle. And then I'm gonna close it up with information. My team and we go to New York every week to study the newest invasive species of ornamentals. That's the box tree moth with a collaboration with the USDA APHIS. We're trying to understand that. We'll talk about that invasive when we get to that point. So I'm going to jump right into what I personally consider, and I was really clear and loud when I started my position here in Virginia Tech. Growers were really concerned about this insect. This is the red-headed flea beetle. On the top right hand corner, you see the picture of an adult, and the adult is the one that causes the damage. You can see a heavily defoliated leaf from an adult feeding. Right? So it's this guy, it's the problematic one. This is what the growers care about when you have all these commercial blocks with your beautiful hydrangeas, your Weigelias, even sometimes roses, hollies. All those are the preferred hosts for this guy. So this guys, when they came to the system, they will do will start as an overwintering as eggs, and they use the potting mix, the potting media to stay as eggs and also hatch for the adults. I'm sorry, the immature. So the immatures are the one they're going to be feeding in that potting mix and feeding on all the roots systems that you have it in the pot, right? Then after that, they're going to close as adults. And usually we expect those adults to be coming. I'm going to show it to you when exactly we expect to see coming. I would assume that it's something similar, but maybe you guys are going to have earlier occurrences of this adults in the system, since you're a little bit souder than we are. Growers are really typically spray twice a week when there is really a hot spot and a lot of pressure from this. So you can start thinking about how much of concern this insect could be for affected growers. So we started working with trying to understand when this beetle was present. So this is for adults throughout the growing season. On the bottom, what you're seeing is the different collection dates. Those are and we did it once a week. And then on top, you have the hydrangea. That's one of the taxa that is preferred by this adult red-headed flea beetle to feed on. When we have the hollies, and then we have the Virginia sweet spire Iteas. These are the one of them, top sell ornamental plants. And unfortunately, this beetle likes to feed on those. And here, the information that we presented to the growers, and we now have a better understanding, is that it is expected to have 3 peaks of populations, 3 peaks of activity for the adults, at least in our system here in Virginia. Eastern Virginia is where we start seeing a peak of the population in late June, another one in late July, and one more in late August, early September. So it takes approximately like a month or so between those peak of activity, so the higher densities of adults that we're going to see in the system. And that helps the growers to timing and actually trying to plan their scoutings, and not only scouting, but also the control tactics that they can apply. So after we understood at the landscape level where these beetles are actively present, now that we are at the plant level, we wanted to understand what was going on, what were their activity. So I have this great grad student. She was able to pinpoint everything and she got this crazy idea to do it at every 2 hours. So you can see here on the graph that is 1 in the morning, 3 in the morning, 5 in the morning, 7, all the way to 11 at night. And we were able to track multiple individuals and see in the field, what are they doing? And the hanging fruit question is like, Alejandro, what should we care about that? If we know when they're active, you can start deciding if that influences your scouting. Like here, for example, 7 in the morning, most of the beetles that we found, they were actually hiding and being sedentary and not moving. So if you go out and scout at 7 in the morning, once you're going to get wet, the dew and everything, but 2, you're not going to see the beetles going up and down compared to when you go and see here at 11 in the morning, or maybe at 1 in the afternoon, where they're all flying, they're jumping, and then even walking around 1 o'clock. They're moving around. So if you have a hot spot, or you want to know if will be a problem, maybe you want to consider going to those specific time of the day in order to take a look at it and see if your crop is infested. Alejandro, I won't be able to do every single block at 1 p.m. Actually, I'm enjoying my lunch. But now you can be a little picky about it, right? See, if you have this information, you can use it in your advantage to try to see if you can accommodate at least those hotspots and those problematic blocks that you want to take a look at it. That was the whole idea behind this daily activity. We also noticed when we were looking at the plant level that this beetle was somehow making the damages were distributed all over the place. So we thought that it was this random, there was some randomness about seeing the damage from the beetles, say on the top of the canopy, or maybe in the bottom, maybe on the young leaves, maybe on the older leaves. And why is it important? Because you want to be mindful about that when you're plant coverage, when you spray, right? So if you want to spray the younger tissue, maybe you will focus on doing a lighter coverage compared to something that it's older tissue where you really, maybe you want to penetrate and go inside the canopy. So what we did is we did no choice and choice assays. So we use hydrangeas as a model, and then we put the beetles and we actually start giving them some choice. And then this one was no choice. So what we did is we have a mature leaves, the same, it was big one. So we cut out a square. So that's the Y bars here. But then when we have the mature leaf and then a young leaf, we were able to track their feeding, the percent of damage for each leaf at 1 beetles, 3 beetles, 5 beetles. Those are the columns. And then when you see a cross, so that will be the rows, it's in 24 hours and 48 hours. So there is some sort of like a linear relationship. And that was expected. That the more beetles you put, the more damage you're going to see. And then the more time you wait, the more feeding you're going to see. But here's what happens. We were able to see that young leaves were somehow getting more feeding. They were withstanding more feeding. So look at this one, for example. It's over 10% in 24 hours when you have one beetle, it is a young leaf. Like if you think about that and then you start making the numbers and the math when every single beetle can be able to do more than 10% of the leaf and all of the leaves. So that's why growers are really looking at this as an issue, right? So we want to corroborate this, and we did a cage study where we actually wanted to look at, now that they have a choice within the plant, what are they going to choose? And they actually going after the old leaves. So then we decided to do this, some sort of laboratory choice assay when we put young tissue and old tissue in the two extremes and drop the beetle at the middle and trying to see how the beetle is going to choose. And at the end, they were actually choosing to go after the mature leaves. We were looking at the literature, and what happened is there are some compounds being present in the case of hydrangeas. There are some alkaloids and other compounds that were present in the young leaf compared to the mature leaf. So maybe that might be driving. We couldn't go further, but this is something that caught our eye. It is some sort of relationship that we're gonna see if there is some preference or not. In the field, you're still seeing all this different type of tissue being fed. You're still seeing the old, you're still seeing the young leaf. So I think we need to better understand this and see if there is any chemical relationships involved, not only just for the fact of being type of tissue. Now, what we ended it doing on top is to see, all right, we got that functional response. So that was this on the left-hand side, that was a corroboration. So now at the plant level, if you have 0 beetles, 5 beetles, and 25 beetles, this is the percent defoliation that you're going to expect every single leaf. So technically, when you have 25 beetles, your leaves are going to get into that 8%, 10% defoliation. But now when we divide it by bottom, middle, and top sections of the canopy, the beetle was not showing any specific location or preference of location, which is interesting because there will be sections of the plant that are going to have younger tissue, mature tissue. So now we start looking at some interactions. So it gets complicated. So long story short, we're going to be able to nerd out and trying to better understand all these different factors between type of tissue, location within the plant. And at the end of the day, the practical implications are about scouting and application of insecticides instead for plant coverage. Right. What we learned so far, red-headed flea beetles are coming inside from the nursery. There were some growers that was trying to point fingers at, hey, my neighbor might have the red-headed flea beetle and they're coming. So we were able to show with a different set of trials that they were basically coming from, and the majority, they were basically coming from the nursery, from that potting mix, that potting soil, right? So something that we are not considering, since the adults are quote unquote fleeing around, moving around and doing all these different things, we might need to start thinking about the immatures in the pot and being one of those, that stage could be the one that we can target for control, right? Multiple applications were justified. I show you how much of the feeding potential a single beetle can do in 24 hours. And that is really detrimental to have most of the leaves being defoliated with a 10% or more. So that's not good. That will not be acceptable. And that will force growers to even prune and delay their own orders in case for affected crops. So this was more reassurance for the growers. Alejandro, we're not just grazing and spraying because of the sake of spraying. It's actually a really tough pest to deal with because of the numbers, the duration within the season, the peaks. We have adults from May all the way to November. So that is persistent, like all these different factors that weigh into the decision for the growers to do multiple applications, right? And then now we're trying to do some remote sensing. We have some tools, but that will be a conversation for a different presentation. All right, so I'm leaving the eastern part of Virginia, where red-headed flea beetle is a problem, and I'm moving to central Virginia, where Japanese maple scale is a huge deal. Here is a boxwood plant on the bottom right that it's been defoliated, and there is some dieback from branches because of the high infestation of this Japanese maple scale. So they overwinter as the females. When you have the Japanese maple scale, your plants will become unsellable. Some of the counties, at least here in Virginia, they're going to stop your shipment if you don't pass inspection. So it can be tricky and problematic as well. So the book says that we expect two generations, one in May, one in August. So now we're digging into the biology and the ecology and the phenology as we did it for the red-headed flea beetle. I think between the two of them, there is still too many gaps in the information that it's not helpful. So we're trying to fill those gaps with this biology, ecology studies that we've done to do. So here is a collage of all the way from the eggs on the top left corner to the letter B, which is the crawler, is the mobile stage. So the first insert for the scales, this is the armor scale, has the shield. It makes a shield, a waxy shield that if it will repel, it's going to, as I said, it's a shield. It will protect them from many applications and things. So it's tricky to manage this. And here you can actually see the newly made waxy shield that they have called the test, right? And then you have this is the first insert under the test with a little bit of magnification. Then you start seeing how they get produced. Those are the adults. And at the end, you have the male and female. Usually females are going to be a little bit rounder. Males are going to be a little bit elongated and broad in terms of the shape what they have as when they are settled and on the infested tissue. So we were curious now that seeing if there is any activity during the winter. And guess what? Japanese maple scale, even though I'm pretty sure, and this is my personal opinion, pretty sure the insects can read, but they don't care about it. The books are written and they said that there is two generations, but Japanese maple scale, like, I don't think I'm gonna pay attention to that. And this is why we were able to see different grad student. She was able to look into this past winter. And what we see is if you go out in the field in mid-January, this is what the graph, the very first panel, when you collect infested tissue, what is going to happen is you're going to see alive early immatures and some immatures. And then we went back to those affected commercial blocks. In this case, will be ornamental trees that have this Japanese maple scale. And those are still present in mid-February. And then all of a sudden, you have this shift within the stages that are present and the affected trees. And now you can see adults meaning that slowly, steady but slowly, Japanese maple scale can have some sort of development. And sometimes we have some mild winters, and that could be one of the factors that we're actually going to start looking into. And they're really in tune with temperature. Therefore, this is what it was expected to see. And now we have some data to corroborate that. That's a little scary because we were trying to go by the book and say, don't worry about it. You can start dealing with Japanese maple scale in May. And now early March, and you're going to have adults. So here, maybe proposing doing some horticultural oils applications, and then they call dormant oils. I'm getting ahead of myself, but that's the rationale behind looking when you're coming out of overwintering. So now we're using the sticky side of the electrical tape as one of the methods to look for crawlers. And why crawlers? Crawlers are the only one stage that doesn't have the waxy protection. So they're really exposed. They're the most vulnerable stage for the Japanese maple scale. So therefore, we want to know when they're present so we can target that. So here you can see that we place those white with the sticky side out, the electrical tape, and then here's a close-up of that electrical tape with all the crawlers in there. So the next question is, now where are you going to place that tape? So far, our data said that the best place within the plant canopy is the middle portion of the plant. That's where you're going to have the higher chances to have crawlers getting stuck on those electrical tape. That's the graph in the bottom right that shows you how we're going to be being able to, and this is the crazy part that we do in the field when we divide it equally all the strata and trying to, that's the fancy part, and then we get and collect the different tapes, and then we analyze. And so far, there's been a consistent trend when you're going to have higher chances to see those crawlers when you put the tapes in the middle portion of the canopy. Again, Japanese maple scale said, yep, the book says that should be started and be a problem in May and August. But guess what? It was a problem in June. It was a problem in July. It was a problem here in August, late July, August, and then start tailing off all the way to mid-October that you start seeing crawlers and so this was eye-opening, some sort of like revelation, not only for us, also for the growers that we're working with. And that was a little scary, but we're still monitoring more. We're still trying to understand the dynamics about scales are so fascinating, but we really need this information so we can propose, and the growers can propose alternatives, and the growers can do an informed decision to when to control. All right, changing gears. Now I'm going to do the pest that is across the state, right? This beetle, ambrosial beetle, and here's the typical damage the ambrosial beetles will do. The toothpaste that is sticking out of the actual tunnels that the female will go and do. And here's another example. Here's the one that we call Crass. Crassiscula is the genus, but Crass is one of the most prevalent species of ambrosial, since it's a complex of multiple species. On the top, you got your manus. If you're asking me, they look the same, but they're not, and all these different things. That's why you have us helping, trying to understand this and do the proper identification. Right? I don't blame them. I'm not going to blame ambrosial beetles. They are attracted to alcohol. Who's not? Who doesn't like ethanol? I'm relaxing. I'm gonna get a beer. They like that too, as animals, and all of us. But then the caveat is that ethanol for ambrosial beetles, which is this tasty and nice drink that you wanted to have, it's because it signals that the trees that are emitting the ethanol are stressed. Therefore, the defenses are lower, and the ambrosial beetle are going to have a higher chance to infect those trees. So here you're enjoying yourself, having a glass of wine. Here you're having a nice pint of beer, and all of a sudden you see this beetle just flying and landing on your beverage, and you say, "What the hell? What's going on?" Now it's just trying to present the information to you that what's happening is they're thinking that's a giant tree that is producing the ethanol, and they can smell it. Even there's a published manuscript from Louisiana that show this is actually the bottoms from the females. They were drilling and tunneling through plastic because this container has gasoline with, I think, was 10 or higher percent of ethanol within mix in the gasoline. And that was the only source that has ethanol emitting from that canister, that plastic. And then the females were able to smell that pretend that was a tree and then go after it and look at it. And their mandibles are so strong that they were able to chew through the plastic and trying to get to that precious price of having that ethanol. Amazing. These guys are amazing. We have multiple years of data trying to understand when we use this 2-liter bottle ethanol with the ethanol baited trap. So here on the right-hand side, you can see the garden hook and the bottle with the lure. And this is just plain and simple 2-liter bottle. You can make it. It's pretty straightforward. And we're trying to provide those solutions to the grower so you guys or any end user won't spend $20, $30 just for a trap, right? So our 20 plus years of data show us that in case of Virginia, and this one will be Eastern Virginia, we are expected to see brochure beetles. In this case, this is just for crassiscus, a peak of activity from April 10th to May 22nd. And then a small peak, some sort of like a little activity. Actually, right now, we're still collecting some beetles all the way from August 15, 16 to September the 20th. Since trapping it's important, and we will start thinking about, and actually, Shimat and I, we are part of this multi-state effort to try to better understand not only the information for brochure beetle, but how we can pull that out together to do the management plan. So trapping is one option. So we were able to test the bottle trap with a low dose of ethanol lure and then with a high dose ethanol lure and do all these different combinations. So long story short, you can see how number of adults, that's what we are seeing here, trap per week can be bumped like automatically when you have that ethanol lure. So it's corroborating what we've seen. And actually, we probably are going to go to look into this and see how maybe at different rates are going to do different responses. And on top of that, the different species that we have might respond differently depending on the ethanol concentration. Right? So now there is another group of studies that we were running right now, not right now during this year and the year before. We're trying to understand how the brochure beetles are flying through the landscape. Why is this important? Because they really want to understand the impacts where the location of these trees could be in relationship to the wooded areas and how the progression of those brochure beetles coming out from those wooded areas to the actual nursery. So here's an example when we start putting the spatial component, the seeing distances and distance from the wooded area, you can see how the traps get the different captures. So here is the dense means higher number of beetles are close to the ones that are yellow and orange compared to the ones that are blue with the lowest capture. So then we're trying to understand how long it takes for brochure beetle to move. And this is in the field. Like we have nursery growers that help us to put all these traps and trying to understand how a brochure beetle moves from in one direction, multiple directions, and how that in relationship to the wooded areas, right? So then we'll have this multiple weeks, and we try to understand how that dispersal activity progresses through time. But here is something that now we're interested because now we want to understand the flight capacity. So how far an brochure beetle can go, and this is called a fly mill, and the teeny tiny thing that you see flying around, that's the brochure beetle. And what happened is with this equipment, we're able to glue those brochure beetles, and then we're able to calculate the number of revolutions that they go, and we do hundreds of these beetles, and getting right now the numbers that we have is brochure beetles can fly within 24 hours, they can fly all the way to 750, 1,000 meters compared to other species or other insects. So the idea is that now we can combine what we see in the field, what their flying potential, and trying to understand if we're going to do mass trapping, how far the traps with the pheromone, the ethanol lures can be placed. Ideally, because now we better understand how far they can fly. All that's just tying together. Pretty exciting. I'm going to change gears. I always, when I have a presentation, I like to bring this up. And it is the integrated pest management, the IPM concept. It still baffles to me that this is not something new. This is nothing that we just came up with it, right? This is nothing that Shimat and we are so prone to propose. It's already been proven that this is a great approach. but it dates back to the 60s. This is not new. But you know what it was striking when I'm putting together, when I put this together, that we have up to 67 definitions for integrated pest managers until today. So that's something that we need to think about that. Surendra Dar, a colleague of Shimat and I, he used to be in University of California. Now he's in Oregon. He puts together in perspective how many layers of information, how many factors can be put together for such a small three-word concept that it actually encompasses so many things. And in this case, the center part is the actual tactics. Then you have to have the knowledge and resources. You need to plan around IPM. But here's what I want to point out. IPM in the core has the research and the extension. And that's what we're doing here today. You're setting on the webinar, you're earning your credits. Sometimes we will sound like a broken record, but here and there, you might hear something that caught your ear, caught your attention, and that is totally worth it. And I think that on top of the business aspect, this is really important. We're all running a business. Agriculture, it's a business. So having that into consideration, also taking care of the environment, it's important. But yeah, we need to fill those orders. We have our clients, and that's also important as well. And then on top, the sustainability. So the long-term effect that IPM can have in the whole system. Do we have an IPM program for ornamentals and nursery? I think the answer is yes, we do. Personally, what I think is there are three pillars. The pillars are cultural control, biological control, and chemical control. We're going to talk about that right now using those previous insects that we were talking about. Cultural control, this is the classical examples that you learn or you already read about. Choose the right variety. Hopefully you watering correctly, you trying to do the fertilization, and it sounds repetitive, but there is plenty of information that shows the correlation or the relationships between all these different cultural factors and the infestations of pests, right? Pruning, sanitation, and all those different good things that you can incorporate it. And I want you to be aware that you're doing a cultural control, that it's actually considered part of the IPM program. These are the underdogs. These are the overachievers that nobody gives them credit, the beneficials, the biological control. So we have this array, this suite of beneficials that they're doing a silent work for us. They're helping us, not in the sense that they can control everything, but at least providing some sort of control, reducing pests, not at the levels that we desire. But start from the top. You got the ladybugs, that's the adult, the immature, that's the eggs. These are lacewings as immatures. This is a parasitic wasp doing a 270, like it's a kung fu movement, trying to insert an egg inside an aphid, right? Then you have the entomopathogenic fungi that actually grows in other insects. Like here, that's an aphid, you have just sporulating conidia from a fungus. These are predatory mites. On the bottom from the left-hand side, that is Aureus minute pyrebug that feeds on thrips. You can see that feeding on thrips. This is a fall armyworm that has the tachinid fly. So those are eggs, and those maggots are going to close and then go inside the caterpillar and then eat the inners. And then on the bottom, you're going to have syrphid flies. There are the bee mimicking ones that actually lay. Those are the eggs, and that maggot is going to eat. They prefer to eat aphids, and they can do up to 20 aphids in a day. I'm impressive. These guys are impressive. I was interested in doing some of that and using technology to promote the implementation of biological control. So now I'm currently using as well. In my previous position, I did that too, using drones to release beneficials. So what we did is we release it, in this case, lacewings, this is the ones in the middle. We release lacewings on multiple crops in order to reduce populations of pests. Here's an example. Kevin Hill is one of my collaborators, still my collaborator. He's located in California, but this is one of the projects we did before I left California. So Kevin is ready to use a drone. This is a 360 video of the drone. That's why it looks like that. And then we have those drums filled with rice hulls and the lacewing eggs. And then the drums start to rotate. And then you can see now how all the rice hulls mixed with the eggs are being dropped from the sky with the drone, and then you can plan your specific crop or the area you want to release those beneficials, and then you can use the drone. So a month ago I did the last release for cotton, and we're releasing lacewings to control the corn earworm. So again, there is now with mainstream and lower costs trying to use these technologies to see if there will be a fit on any IPM program in different and multiple crops. But long story short, insecticide are the one that we use the most. And I'm going to speed up a little bit. So pesticides are the one we want to use. We're interested to see efficiency. So we do all these different laboratory testings. So here, the take-home message is that different active ingredients will have different times when you're gonna see mortality. For example, you see the different colors, the dotted line, and all these lines. So these lines have killed the aphids faster than the other ones. But at the end of the day, after 4 or 5 days, you're still gonna get the 100% mortality, even though you didn't see that at the beginning for these 2 insecticides compared to the other 3. Headed flea beetle, we tested multiple insecticides using bioassays in the lab. We were able to find the ones that were really efficient. We took that to the field, and that's where things started to falling apart. We saw that a lot of variability when we use specific individual plants. Locally, when we look at the damage per leaf, all the insecticides were able to lower, especially let's say a celebrin, was able to lower the percent defoliation per leaf, but it still was defolition. So I think something that happened is the overhead irrigation every day plus the lack of adjuvant must have influenced some of this results. So we're in the process to repeating all this. Something that Shimat and I, we've been working, it's trying to understand how drenching and targeting the immatures in the pods could benefit the overall management for the red-headed flea beetle. So here's an example when we drench using different insecticides. Here, for example, Tetrino, Expire, even Mainspring, you can see how we lower the defoliation compared to the untreated and some of the plants that were treated with Altos and this Apta, which is the same active ingredient, right? Busiest line, long story short, the line is the 50%. This is for Japanese maple scale. The panels are 1 day after treatment, 7 days after treatment, 15 days, 26 and 32 days after treatment. And everything we can see is, here's what we're still trying to understand this, but 7 days, you can see some mortality, but actually when you start to picking up some mortality with all the different insecticide, it's at the 2-week mark. And then the 3-week mark, you're going to see passing the 50% and at least getting to past the 80% mortality for that. So that was a really great learning opportunity for not only us, but the growers to see how these guys can perform under laboratory conditions. We are still trying to look for alternative to pyrethroids. Pyrethroids are one of the most chemical used for ambrosia beetle. So pyrethroids are the ones that have the arrow. We have 7 days, 14, 21. So, weekly time frames that we collect data in number of attacks and what we call bolts or pieces of wood that we lure with ethanol, and then we coat it with the insecticide. At the end of the day, there's a lot of inconsistencies. Nothing can outperform so far that we tested can outperform the pyrethroid, which is we're still hopeful. We're gonna keep pushing and see what we can find as an alternative to see if we can rotate pyrethroids in order to prevent ambrosia beetle damage. All right, this is the last section of the talk. I'm gonna go ahead and talk about the boxtree moth. This is the latest invasive in the United States that feeds on boxwood. And you can see here in the picture that is a boxwood that we scouted in July 11, and 14 days later, it was completely gone, defoliated by the caterpillars. This is pictures from Buffalo, the Buffalo area. So that's Niagara Falls in New York, upstate New York. These are boxwood hedgerows that are completely gone. And what is people doing? They're taking it apart, cutting it, and put it on the side of the road for the city to pick them up because they give up. The caterpillars are so voracious that in a week and that in 2 weeks, it's completely gone. So box tree moth It's moth. It looks really pretty, but it's devastating. So that's the eggs. Those are the teeny tiny early instars. This is the full grown caterpillar with the dark hair capsule and all these lines and dots and the hairs, and that's the pupae. So native from Asia, came to Europe in 2006, and then moved to Toronto, Canada, so which is North America, back in August 2018. We expect to have 2 to 3 generations, and they overwinter as a third instar. Right now, the epicenter for us for the United States was Buffalo, New York. Then he moved to Michigan in November 2022, and then they moved to Ohio in June 2023. And the latest report of the presence of box tree moths, BTM, in the United States is Massachusetts in September. This early September, we got an alert saying that it was detected over there. So you can see it already started spreading out in the United States, and this is concerning. Here is showing you some of the phenology. On top are the adults, in the middle are the early instar caterpillars, and then on the bottom are the late instar caterpillars, so the bigger caterpillars, right? So the adults are expected to peak during July, the presence, and also right now they're coming out of their peak as we speak, right? They're coming out for that second peak, and then all the caterpillars are getting ready to overwinter and go to the next year, so that will be 2024. We are looking at the biological control, so the beneficials. They're present in the system. Unfortunately, beneficials are still naive. They don't recognize BTM as a prey. So that means that we run some correlations, and so that doesn't matter how many beneficials you have. If you have BTM with the caterpillars, they're still going to be there, but there's some hope. Actually, last week, one of my team members was able to recover this parasitoid. It's an ichneumonic parasitoid, recover from one of the BTM pupa that we're collecting. So because we're collecting groups of pupa every certain times, and finally we were able to collect the parasitoid. So it seems that now the beneficials are learning that box tree moth, it's a new prey item, and they're going to start going after. This is an airwick looking for a caterpillar. You can see all the damage, and the airwick just like trying to feed on something there. So USDA has suggested to use diamides, spinosyn, and BT, Bacillus thuringiensis, that one of the insecticide of choice to control BTM across the United States. Homeowners are trying to do their best to go to the garden centers and get something and spray something to control. We are, as we speak, trying to use mating disruption, so using the sex pheromone on dispensers to confuse the males. And actually, we do it in an urban setting, and it's looking promising. I don't have the data here, but so far, we have lower captures and lower numbers of caterpillars in the areas of the towns that we put the pheromone in it compared to the ones that were not treated. So this is looking really good. Last but not least, I'm also doing some turf, and I'm using light reflectance to try to pinpoint where the turf is infested. In this case, with Anno blue rust weevil, we have all these different profiles, the light reflectance to see when the beetles are present as adults and immatures, how that responds for the turf. And we also are using remote sensing, in this case, using some of the light reflectance from the out of play areas to trying to locate locations where they can serve the pollinators. So we want to see if there is any specific light reflectance coefficient. In this case, it's called, you can see here, SIPI, and PSRI, actually. PSRI is the one that has the darker values. The darker is the higher correlation between the presence of pollinators and that calculation of the light reflectance index that we can use. So we can map out entire fairways or whole golf courses in order to pinpoint where those out of play areas can serve the pollinators if a superintendent is interested to promote that in the near future. With that, just my final slide, the red-headed flea beetle, we can use it as an example of an emerging pest. It's a critical defoliator, and we're working really hard to provide those answers to the growers. Japanese maple scale could be a quarantine pest. What I mean by that is your shipment can be rejected if you're going to do some sort of certification. Some states will require that certification on an inspection. So if you have GMS, it's going to be a pain in the neck, pardon my French, but it will be painful. And you have to have some actions and correction actions to pass those inspections. Ambrosio beetle is still persistent, came into the United States back in the 30s, and all decided to stay. So we need to learn and how to put all the information together. Remember that the fly mill and all those cool things, put it all together to propose a management strategy. Box tree moth is the new headache right now, is the new invasive species. USDA, ARS, and basically aphids is working really hard to help us and how we want to move forward as an industry in order to do certifications, quarantines, and treatments. And then remote sensing, I'm using remote sensing of one of the methods and ways to detect different pests and different indirectly, because what we want to collect is the stress of the plants and trying to correlate that with insect pests. So at the end, IPM, I know I'm going to sound like a broken record, but IPM is the way to go. It actually has been proven that it will help us to reduce those economical and also the environmental risk. And basically what we have right now, the chemical control is the top one most used, but we definitely have those alternative controls that we can employ in order to improve our IPM system. With that, that's a picture of my house. I wish that's a picture of my house. That's a beautiful, like how peaceful and beautiful that view is. This is how our industry, the green industry, represents and all those benefits to us as a society. So that's what we care deeply about, protecting this industry and serve the industry in many ways. So that's my name. My email is right there. Please feel free to contact me at any time if you have further questions, and I will be happy to, if there is any, or if you guys are interested to talk a little bit more about anything that I presented. With that, I call it good.
Thank you so much, Alejandro. It was an excellent talk. Really appreciate it. And for the audience, this is time for questions. If you have any questions, use this Q&A box there, and you can type your question, and I am more than happy to read it out, and Alejandro can answer. So we still have some few minutes to go, so we're good. So Alejandro, maybe there's a few minutes saying the thing about the question. They had to decide that, and so
it will be overwhelming. There's a lot of diversity, but our systems, we are so diverse. There is multiple taxa and multiple things. So the question is, where do you start? What are you going to focus on? And locally, the growers and the economic impact of the specific insects are the ones that drives our programs, right? So in this case, red-headed flea beetle, Japanese maple scale, and ambrosia beetles are the top ones that we really need to work on. And that also serves, like ambrosia beetle, for example, it's a problem for a landscape company. Like I have so many calls, people say, Alejandro, I have a magnolia tree or this here, and I have a nice customer here, and they're asking me about all these different things. So it's good to have that information and explain that when the ambrosia beetle is expected to fly, what is happening in the system, and all these different things. So I think overall, that's probably why they might be shy and they don't want to ask questions. It's too much, too confusing.
So I have a question regarding box tree moth. So it's a new invasive up in north now. So it looks like after introduction in 2021, it's spreading and it's on four states, right?
Yep.
Can you explain how they are moved around, how they are dispersed from the origin? Is there any clues, any information we have on how they are moved around and how they're spreading?
Bro, for the anecdotal observations we have, Shimad, it's when you go to Niagara, that Buffalo area, you have Lake Ontario across, and they are, what I'm trying to say is really strong flyers. So the upstate New York is a fragmented landscape. And fragmented is like you have one or two boxwood plants here, and then you drive for 2 miles and you have another set of boxwood. And then you're going to find caterpillars in both of them. So that means that the moths are flying around and they're really strong flyers. So I think one of the main drivers to do a local expansion year by year is because they're strong flyers. Also, there is a lot of movement, right? So that's why quarantines and expansions are really important, even though they're an appending the neck for the growers. But that helps us to slow the spread. And our work with mating disruption could be another layer. So all the nurseries that are affected can put that pheromone mating disruption program, and then they're adding that control, right? So you reduce the chances, the risk for distributing egg masses, caterpillars, and anything that might be missing in the certification.
Is there any detection from nurseries thus far? Because that's what I was wondering.
Yes, nurseries that are in the Buffalo area, are already infected and they are spraying BT, Spinoza, and sometimes some pyrethroids, but mostly BT to control their caterpillars.
Looks like we have a question. Maybe I could answer that. Is the Japanese maple scale present in Georgia? So I would just say yes. The answer is yes. I have seen this problem in upper part of our state. I don't know specifically saying which region, but there are nurseries where I have seen problems with Japanese maple scale issues. And where I have seen that's mostly during early summertime. And either if you're aware of crepe myrtle box scale, it's very similar looking. But not exactly the same because box scale, the females, the herd scale, there is no sexual dimorphism or anything like that. But with the Japanese maple scale, you'll see that comma shape structure that Alejandro showed. And we have seen that in Georgia. It's not really widespread in Georgia, but it's there. And wherever I've seen is from nurseries. So it's there. Another question I have, again, going back to box tree moth, because that's one thing we are already good.
Yep, absolutely. I'm happy to talk about box tree moth. Yeah. So what's the question?
Is it only attacking box tree, or is it going outside other plant species?
So this is being a back and forth. So boxwood is the primary and the most preferred host. There is some evidence in Asia that there is another type of plant, Euonymus, that might have, and that was included because of literature for the United States. But there is no evidence in practical, and that what we've seen that actually feeds on. So there is some conversations about how that it's holding up through for the new invasive range. So far, what we see, it's boxwood, it's the primary, so Buxus is the primary genus. The box tree moth is actually feeding on, and it's a single plant taxon. Right?
Boxwood is not native to US, right? Is native to.
There are some native ones in Puerto Rico. There's some other natives, but I believe it's mostly European. There's some Asian, right? That's the area when that genus, based on my understanding, it was originated.
So they suspect the infestation is going to be restricted to urban areas, you think, because that's where the landscapers plant a lot of boxwood?
Yeah. So, for example, upstate New York, it's a prime example of they love boxwood because in the winter, the only thing that's going to be green is boxwood. So they really want to have the boxwood. So therefore, they have the host and they're moving around. So here in Virginia, Williamsburg, for example, it's a prime example of an area that it's full of boxwood. So if something happens and it lands it in here, we're going to have areas where are more prone to compared to others. So yeah.
Great. Looks like we don't have any more questions. And for the sake of time, I will call it off. And I want to thank you, Alejandro, for an excellent presentation going through several major pests affecting nurseries and also landscape, I would say.
Yep, absolutely.
Appreciate and for your time to participating in this webinar. Okay.
All right. Sounds good. I'll stick around. Thank you.